Welcome to my channel, and today we are going to be looking at microscopes. Before I begin though, don't forget to hit that subscribe button, share this video, smash that like, and don't forget to hit that bell icon. I also have these nifty timestamps, so if you want to skip to a specific bit, do so. Now before we delve into microscopes, there are a few key terms that we need to understand. We have resolution, we have magnification, and we have field of view. Resolution is the ability to distinguish between 2 points on an image. Here is an example that you guys might be familiar with. Here is an image which is a low resolution image, and then see what happens when I increase the resolution. As you can see, it's much easier to distinguish between different objects in the image. Let's show you another example of low versus high resolution with one of my favorite video games of all time, Elden Ring. If you notice, at low resolution, it's a bit hard to distinguish between the different blades of grass, and the emblem on the cape also does not look very clear. At higher resolution, it's easier to see the different blades of grass, and the emblem is much sharper as well. As you can see, higher resolutions give us more detail, and a key word to use is that the image is sharper. Now, magnification is the ability to increase or decrease the image size of an object relative to its actual size. You guys are familiar with magnification when you use your phones or your cameras to take pictures. When you zoom in, you are increasing the magnification, and when you zoom out, you're decreasing the magnification. Now, a key thing to remember is that by reducing the magnification or switching to a lower magnification, we actually increase the field of view. Now, the field of view is how much of a sample we can actually observe, the extent at which we can observe a sample, whether it's an object or whether it's an area. To demonstrate this, I'm going to go back to another favorite game of mine, Guild Wars 2. Here at a higher magnification, you can see that we can focus on this practice dummy. But when we go to a lower magnification, you see that we can see more of the area and the environment around the dummy. So now at a lower magnification, we have a greater field of view. Now all this talk about microscopes and no microscope images, what a tragedy. Here we go. Here we have a lower magnification image of a blood sample. You can see all the red blood cells and some white blood cells inside. Now at a higher magnification, we can look closer at a bunch of those cells, but we've lost sight of the surrounding cells, and that is a reduction of the field of view. Now when it comes to microscopes, there are 2 types of microscopes we need to know about. There is the electron microscope, and then there is the light microscope.
Let's get ready to rumble!
Now, electron microscopes have high resolutions, they have high magnifications. In fact, they are so good that you can actually look right inside an organelle of a cell. Unfortunately, electron microscopes create grayscale images. Any living sample that will be looked at in an electron microscope will probably die, and electron microscopes require specialist training to use. And of course, there is the elephant in the room. Electron microscopes are very expensive. Let's have a quick look at what an electron microscope might cost.
What the hell?
Damn! I can't even afford a light microscope for this video. How am I gonna afford an electron microscope? Now let's look at the different components of a light microscope and what are their functions. Firstly, there is the eyepiece. This is of course what we use to look at our sample. However, an important point to remember is that the eyepiece itself has a magnifying lens inside, usually of a ×10 value. This little platform here is called a stage. This is where you place your sample or your specimen slide. This right here is the light source for the light microscope, which is going to allow us to see our sample. And just like the aperture on a camera, this can be adjusted to let more or less light through the sample. Then there are the objective lenses, a selection of lenses with different magnification values. But instead of saying different magnifications, we might use the term power. So a high power lens is a high magnification lens. The fine adjustment knob is used to focus on the image and clear it up if it's a little blurry. It's very similar to the fine focus function that you might have on your phone cameras. Now the coarse adjustment knob is a bit different from fine focus and a bit different from the job of the objective lens. Some people even call it the coarse focus, but it's not quite accurate. And let me show you why that is. You see, when you use a camera, there are usually two functions available to you. There's the zoom function, and the microscope equivalent of that is the objective lens. And then there's the fine focus. The microscope equivalent of that is, of course, the fine adjustment. There is, however, no coarse adjustment equivalent on a camera, because the coarse adjustment knob actually brings the subject that you're looking at closer or further away from the objective lens. Simply put, the job of the coarse adjustment knob is to adjust the position of the stage. Now, even though while using a microscope, you might look at a host of different samples and types of cells, but one of the key practicals that you're going to do is looking at an onion skin cell. So let's get chopping. Now, the onion cells that we are going to look at under the microscope are usually onion skin cells or epidermal cells. The tears you see are a combination of me chopping onions and also realizing how sad my life is. We chop up the onion, and on one of the chopped bits we use tweezers to peel off one of the outer skin layers of the onion. Now the key thing to remember here is that we're not actually using the more rough or crusty looking outer skin that we find on an onion. We actually remove that and then we take one of the more clearer looking ones, which is why we are going to be using stains to essentially color it in to make it easier to look under the microscope. Once we have our onion skin, step 1, we drop a drop of distilled water at the center of a microscope glass slide. The distilled water makes it easier to place the onion skin onto the slide and stretch it out. Step 2. Place the onion skin onto the glass slide. Step 3. We will stain the samples using special dyes. The two options we have is iodine and methylene blue. For the onion skin cells, we will be using iodine, which gives it an orange-brown color. After dropping 1 or 2 drops of iodine onto the sample, we then move on to step 4. Once the sample is stained, we then place a glass cover slip on top of the sample. However, we place the edge of the slip first and then slowly lower it down onto the sample. This is to avoid air bubbles from forming. Otherwise, if you look under the microscope, all you will see is air bubbles. Now usually when you place a cover slip onto the sample, it might push out some of the liquid from underneath it. So step 5 is drying up any excess that's been pushed out on to the rest of the glass slide. Step 6, we place our sample slide onto the microscope stage. Step 7, we switch to the lowest powered objective lens first. Usually, if you're in a school, the microscope's lowest power is ×4. And the objective lens might have a red band around it. Step 8. Using the coarse adjustment knob, move the stage up or down until you can see the sample through the eyepiece lens. Step 9. Use the fine adjustment knob to finely focus and sharpen the image. And finally, step 10. Once you are happy with your results, switch to a higher powered objective lens and then make a new observation. Just be careful though. Some of those higher power lens can actually break through the glass slide if you're not careful with the coarse adjustment knob. Now instead of onion skin cells, we can also check out our cheek cells. So we use cotton swabs to scrape the inside of our cheeks and then we smear the sample onto the glass slide. Now due to the saliva present, we don't actually need to use distilled water in this one. And this time around we use methylene blue to stain the sample. Once the stain is in place the cover slip and Bob's your uncle. For reference, this is what a typical onion skin cell sample might look like, and this is what a typical cheek cell sample might look like. Now that's the end of Microscopes Part 1. Be sure to watch Microscopes Part 2 to see the maths side of things. Thank you very much. Don't forget to hit that like button, share this video and subscribe. And if you can, please support me on buymeacoffee.com/insanmusic.